I'd like to thank Sean for the invitation and for the opportunity to be Nancy for a moment. So I'm going to tell you about some of the work. Can you hear me okay? About the work we've been doing in Newcastle to develop IVF-based techniques to try and prevent the transmission of mitochondrial DNA disease. To just give an outline of the talk, so I'm going to start by introducing you to the main players, and John has done some of this already. And then we're going to talk about about the inheritance of DNA, where we get it from, the reproductive consequences of mitochondrial DNA mutations, and IVF-based techniques to reduce the risk of mitochondrial disease. And in that, I'll be talking about what we can currently offer, and also about the new techniques that are in development. So here are the main players. So this is DNA, and DNA is organized into genes which encode all of the instructions required for life, required for our cells to function. And our body is made up of many different cell types, and the cell is a basic unit of life, contains a nucleus which sits in a soup called the cytoplasm. And the nucleus contains nuclear DNA, and it's known as the control center of the cell. The nuclear DNA is packaged into long linear molecules, packaged into chromosomes, and it encodes most of the genetic instructions required for our cells to function. Now, in the cytoplasm, we've got mitochondria, as we heard from John, and these are known as the powerhouses of the cell because they produce the energy. And the mitochondria also contain DNA in small circular molecules. It encodes only 37 genes which are involved exclusively in energy production. So we get our nuclear DNA. Well, let's start here with the fertilized egg. This is how we all start our lives, as a single cell. And the nuclear DNA contained in this, there are two large nuclei here called pronuclei, and they each contain a copy of a chromosome there's a male and female pronucleus. The female pronucleus contains chromosomes that we inherit from our mother. So a single copy of each chromosome, 23 from mom, and we get 23 from dad via the sperm. And then when this embryo begins to divide, these chromosomes combine, so all of our cells contain 2 copies of each chromosome. So this is the nuclear genome, and the nuclear genome here is unique to this fertilized egg. No other cell or entity has the same genetic composition as this fertilized egg. So the mitochondria, as we say, also contains DNA, and the mitochondrial DNA is inherited exclusively from our mothers. So all of the mitochondria in the fertilized egg is inherited from the oocyte. And I use these terms kind of interchangeably, oocyte or egg. Now the sperm also contains mitochondria that live around here because it needs this to give it the energy to swim to the egg. But once fertilization occurs, the sperm's mitochondria are killed by the egg, and we just get the mitochondria that are inherited from the egg. And human oocytes contain between 200,000 and 500,000 copies of mitochondrial DNA. So this fertilized egg will undergo a number of cell divisions during the first 5 or 6 days of life, and it eventually will differentiate to form what we call a blastocyst. And this is an important developmental milestone because these embryos need to develop to the blastocyst before they can implant. And the blastocyst contains an outer layer of trophectoderm cells, which will become the placenta, and an inner cell mass here, which will contribute to the fetal tissues. And an important point to make here is that a tiny number, the blastocyst contains altogether between 100 and 200 cells, but a tiny number, we estimate someplace between 10 and 15, will actually contribute to the fetal tissues. So during these early days of development, the emphasis is mainly on the production of extra embryonic tissue to allow the embryo to implant. And then just a very small number of founder cells will give rise to the fetus. So what happens then if we inherit mutations? We can inherit mutations in the nuclear genome or in the mitochondrial genome. And DNA mutations are like spelling mistakes in the genetic code, and if that spelling mistake is bad enough, it will affect the structure of proteins that they encode, and that can make it difficult for the cells to function and can result in serious disease. So mutations in the nuclear DNA have a wide range of adverse effects on human health and generally manifest as developmental defects. And mutations in the mitochondrial DNA affect energy production, and as we heard in the last talk from John, that this causes a wide range of devastating disease. So if the oocyte contains mutated mitochondrial DNA, which I show here in red, then that segregates between cells. So during this pre-implantation phase, when we get to the blastocyst stage, during each round of cell division, the nuclear DNA will replicate itself. So by the time we get down here to the blastocyst stage, each cell contains a new copy of nuclear DNA. However, the mitochondrial DNA does not increase during this division. And so if we have mutations starting in the egg, these mutated copies of mitochondrial DNA will segregate randomly during development, such that by the time we get down here to a small number of cells that give rise to the embryo, we may end up with different mutation loads between different cells, which can result in different mutation loads in different organ systems. And this effect is really, really marked when it comes to the female germ cell lineage. A woman who carries a mutation, a mitochondrial DNA mutation, can end up with 3 reproductive outcomes. Her egg might contain no mutations. It might contain a mix of mutated mitochondrial DNA and normal mitochondrial DNA, or it can have 100% mutation. And that can occur in the same woman because of the way the mutations segregate during early development. So this condition of a mix of mutated mitochondrial DNA and wild type is called heteroplasmy, and then homoplasmy is when all of the mitochondrial DNA carries mutation. And the severity of disease depends on the mutation load. So in this case, there is a high risk of disease when all the baby born as a result of fertilization of an egg that is homoplasmic for a mutation has a very high risk of being seriously affected. Whereas with the heteroplasmic, when there's a mix of wild type and mutated, you can have the severity of disease depends on the ratio of wild type to mutated mitochondrial DNA. So the reproductive consequences are, in heteroplasmy, the mutation load can vary between oocytes. That makes it very, very difficult to predict the risk of mitochondrial disease in children born to that woman. In homoplasmy, if there's a woman who's homoplasmic for a mutation, all of her eggs will be affected. However, even within the homoplasmic group, the severity of symptoms can vary between individuals. So it makes women who carry mutations face very, very difficult reproductive choices, especially in this group where there's a great uncertainty about whether a child would be affected or not. So the question of whether assisted reproductive technologies can be used to reduce the risk of a woman transmitting these mutations to her children. So there is a procedure which we can currently offer, and this is called pre-implantation genetic diagnosis. And this has been in use for a number of decades now to prevent transmission of nuclear DNA mutations. So it's well established, and it's been on offer for some time, and many hundreds of thousands of babies probably by now have been born as a result of it. But I should say it's only recently been applied for mitochondrial DNA mutations. So the process involved in this pre-implantation genetic diagnosis is that a couple will undergo IVF treatment. They will use the use of drugs to stimulate the ovaries to produce multiple oocytes. Then we get multiple embryos from those. And then we let the embryos divide for 3 days in culture and remove either 1 or 2 cells from here, send them off to the mitochondrial lab, and have them analyzed so that we can tell the mutation load in these cells. Right? Cells can also be removed from the blastocyst stage. We can remove a little biopsy of cells from here and have those tested as well. And in this way, we can identify embryos with high mutation load, and these are excluded from use in treatment. Now, questions about the pre-implant, about the use of PGD for mitochondrial disease are the cells that we remove, are they representative of the entire embryo? Because as I mentioned, these mutated mitochondrial DNA can segregate randomly between cells during embryonic development. And then, is the mutation load in the cell removed here, is it predictive of the levels born in the newborn, levels found in the newborn baby? So it's early days with this procedure. It's an offer in just a limited number of clinics around the world. But it seems that these cells that are removed are generally representative, and that they are predictive of the small number of babies that have been born so far. So to summarize in PGD for mitochondrial disease, the evidence to date indicates that PGD provides a promising risk reduction strategy for affected families. However, and this is a really important point, it's not appropriate for all women with mutations. It's obviously not appropriate for women who are homoplasmic for a pathogenic mutation, because all of their eggs would be affected. They would produce no normal embryos. And also women who are heteroplasmic but do not produce any embryos below the threshold for disease. And on the basis of what we see so far in Newcastle, about 20% of women would fall in this category. None of their embryos are suitable for replacement because the mutation load is high. So the question is, what can we do for these women? Are there alternative strategies we could use for women who carry high mutation loads? And an obvious thing was, can we uncouple the inheritance of nuclear DNA from mitochondrial DNA? Now, there are too many copies of, too many mitochondria in there to hoover them all up. So by far the most sensible thing to do is to actually move the nuclear DNA. So we transplant the nuclear DNA, and in effect, by doing that, you swap the cytoplasm, which includes the mitochondria. So it's mitochondrial replacement by nuclear genome transplantation. And the concept is that you would create this reconstituted zygote or fertilized egg containing the nuclear DNA of the affected woman and her partner, and the mitochondrial DNA from a healthy donor. And there are a number of approaches currently being investigated for this. One is that we transplant the nuclear DNA before fertilization. The other is that we do it after fertilization. The after fertilization one is called pronuclear transfer. And you can see here that you can easily see the nuclei. A complication with it here is that the nuclear DNA here is not actually contained in a nucleus. It's sitting on a spindle waiting for the egg to be fertilized. And after fertilization, the egg will lose half of its chromosomes into another tiny little structure here called the polar body. And therefore, we cannot easily see what we're doing here, because you need, and I'll explain in the next slides, how you need a special form of microscopy to actually be able to visualize the apparatus there. So the transplantation of the nuclear DNA by pronuclear transfer, the idea in the context of clinical treatment, which isn't on offer yet, but this is just for the purposes of illustration, you would get a donor oocyte and a patient oocyte, and you fertilize them, and then you let them form their pronuclei, and then you remove the pronucleus from the pronuclei from the donor oocyte, and you keep the cytoplasm with its mitochondria. And here in the patient oocyte, you fertilize it, and this time you remove the pronuclei and you fuse them with the cytoplasm from the donor oocyte. And this gives us this reconstituted zygote. Now, that was first reported in 1983 in the mouse, in a paper in Science by McGrath and Davor-Salter. And then we did a proof of concept studies, which I'll tell you about in the next slides, which were published in 2010 in the human. The other approach, as I mentioned earlier in the last slide, was that you can transfer the nuclear DNA before the egg is fertilized. And so we've got to use this polarized light to be able to see the spindle here, and it just kind of flicks at us, and we take that out, and we just make the assumption that the chromosomes are here sitting in the center of the spindle, which as women get older isn't always the case. But so this spindle is the same concept again. We get the donor oocyte, the patient oocyte, we remove the nuclear DNA from the donor, and then you just have the free cytoplasm, and then you switch the spindle from the patient oocyte into the donor oocyte, and then you have this reconstituted egg, which you fertilize, and you end up with the same end product as you had above. Now, this was published first, proof of concept studies in the monkey in 2009, and then the same group published it in a human in 2012, and then another New York-based group published at the same time. So this has been mainly taken forward in the US, and this is the approach we've been exploring in Newcastle. There are clear differences between the procedures in that for this one you've got to create an embryo in order to do the procedure. Here you can work with unfertilized eggs. But here you can see the nucleus, here you can't. So there's for and against, and I think in the end the one that's used for treatment, maybe both will be used for treatment, but will depend on a balance of what is the efficiency and what is most effective in reducing the risk of disease. Now, as I mentioned, pronuclear transfer was pioneered in the mouse, and it was proven to be compatible with development in mice. And then in a later study in Japan, where they had a mouse with a mitochondrial DNA deletion, it was missing part of its mitochondrial DNA, they were able to show that they reduced transmission of that deletion by performing pronuclear transfer in the mouse. So our research questions when we started doing this work, we were asking, is it feasible in the human? So is it technically feasible in humans? Is it compatible with onward development of embryos? Really, that means can embryos develop to the blastocyst stage where they could implant? And does it have the potential to prevent transmission of disease? And that really depends on how much mitochondria is carried over from what would be the patient oocyte. So if you see when we take out this pronucleus here, it's surrounded by cytoplasm, which may contain mitochondria. And then if this mitochondria accounts for a significant proportion of the total mitochondrial DNA in the reconstituted zygote, then there may be a risk of some symptoms. So these were the questions we set out with. And getting started with this, one of the challenges was we needed fertilized eggs, but we didn't have a ready supply of those for research. So what we did was we're attached to an IVF clinic. We used abnormally fertilized eggs. So during normal IVF treatment, about 18% of oocytes fertilize abnormally. And that's usually because an extra sperm gets in, so you've got 3 pronuclei instead of 2, or you just end up with 1 pronucleus for various reasons. So our starting point was to use these abnormally fertilized zygotes to see was it technically feasible. So the first step is to see, can we remove the pronuclei? Now these are very, very big structures in the human. And you've got to treat this zygote, so every cell, including the fertilized egg, has something that is equivalent to our skeletons to give it structure. We call it the cytoskeleton. It's made up of 2 components, actin and microtubules, that gives the cell rigidity and structure. So you've got to inhibit the cytoskeleton in order to be able to remove the pronucleus. Then you just sort of pinch off the pronucleus when the cytoplasm is all soft and floppy. Then the second step is you've got to fuse them back in. And actually this was the most challenging part of getting started, because in the animal work that had been done, they were using electrical fusion to make those membranes fuse. But human zygotes do not tolerate that very well. So we had to go back to an older way of doing things, which involved using an inactivated viral protein. So it's using the same technology as the virus uses to get into our cell. The virus is inactivated, but we keep the envelope protein that enables it to fuse with cells. So we can just put a little coating of that protein into this what we call the karyoplast, that's the pronucleus surrounded by cytoplasm, and then we just leave it there and it fuses very quickly, and that turned out to be really efficient and didn't cause degeneration of the zygotes. So that was a big step forward in our sort of getting off the ground with this. So now that we could take the pronuclei out and fuse them back in, the question was, can they develop to the blastocyst stage? Would these embryos ever be able to implant? And so this is an image of a blastocyst created by pronuclear transfer, and that's another one where it begins to hatch from its out of the through the shell that it's formed in. So this is blastocyst development from abnormally fertilized zygotes. So the controls have about 17% development of blastocyst stage. That's much lower than for normally fertilized. We're looking at 50 to 60% development of blastocyst stage. Now the ones that we manipulated and did pronuclear transfer on had a development rate of 8%. Now these were small beginnings, but it at least made us feel that, you know, it's worth looking at a bit further. And the question then of whether we were reducing the of what level of mitochondrial DNA we were transferring with the pronucleus had to be addressed, and this is done in Doug Turnbull's lab. We let the embryos develop, and then we send the cells to his lab, and they do mitochondrial DNA analysis so they can distinguish between the two mitochondrial genotypes. And when we were starting with this, we were taking quite a lot of cytoplasm, and the level of carryover was so this clear area here is the pronucleus surrounded by cytoplasm, and if you've got a lot of cytoplasm, you end up transferring a significant proportion of mitochondria. And when we did our measurements initially, it was in the region of 8 to 15% carryover. So in the total zygote, about 8 to 15% was accounted for by the mitochondrial DNA that went across with the pronucleus. Now, we optimized that further, and we got it down to about 2% in the proof of concept work. So we felt that this was low enough to prevent disease, because disease symptoms, severe disease symptoms for most mutations kick in at around 60% mutation load. So to summarize the proof of concept work, we showed it was technically feasible, compatible with development of the blastocyst stage, and we had low levels of mitochondrial DNA carryover, which gave us an indication that this could be used to prevent transmission of disease. So in ongoing research, which is funded by the Wellcome Trust, we're asking, are pronuclear transfer blastocysts normal? We're refining the procedures to maximize the chance of pregnancy, that's to maximize the development of blastocyst stage and to improve the quality of those blastocysts, and to further minimize mitochondrial DNA carryover. Now, this research requires a source of normally fertilized eggs, because as you saw in the previous slides, these abnormally fertilized ones do not develop to the blastocyst stage with any credible sort of rate, and also they're abnormal to start with, so they're not a good baseline from which to study whether a blastocyst is normal or not. So we had to move to a situation where we were getting eggs donated specifically for research and fertilizing them specifically for this research as well. Now, so where do we get those eggs from? We get egg sharing for research. That's women who are already undergoing IVF treatment donate half of their eggs in return for a subsidy towards the cost of treatment, and that subsidy comes from research funds. However, most of our eggs these days we get by altruistic donation, and donors are reimbursed for their expenses up to £750, and that has been allowed in the UK just since 2011. And really, this is where we get most of our eggs from these days, and we get a lot of interest. However, only 5.5% of women who express interest eventually donate their eggs. There's a lot of fall off. People are very well intentioned, then they realize what's involved and they can't do it, or else they start off and it's just, I mean, it's an arduous procedure donating eggs, so only 5.5% of them actually come through. So using these normally fertilized eggs, we find that almost 90% of them survive the procedure, And then our blastocyst development with this when we transfer these pronuclei is equivalent to controls close on up to 50%. So in terms of blastocyst development, the procedure is actually working better, much better than I thought it would. And then when we look at the quality of these blastocysts, so when we look at quality of blastocysts, we're looking to see, has that blastocyst expanded nicely? Is it nice and big? Does it have this nice inner cell mass here? And does it have a nice trophectoderm? And so without this component, without the trophectoderm, it won't implant very well. Without this, you won't get a pregnancy that will give rise to a fetus. So we grade them according to these criteria. So in effect here, anything that's green is good. Right? These ones with nice inner cell mass, nice inner cell mass, nice trophectoderm, they're green. So when we look at our pronuclear transfer ones, we're looking at the quality is about equivalent to the controls. And just to show you that these quality grades actually mean something, these data come from the IVF clinic. We do transfer of single blastocysts, and when we look at implantation following transfer of a single blastocyst, we see that these ones that are graded A have more than 50% implantation rate, and then it goes down progressively as the morphology gets worse. So the green ones, the ones that are quoted in green here, have between a 35 and 50% chance of implantation. So we're reasonably convinced, and the numbers are very small here, I'm sorry, that's, we have substantially extended this, and it remains the same, and we are reasonably convinced that we're creating blastocysts that have a good chance of implantation. And then we're in the process of doing further studies on those blastocysts to see how they compare with embryos that are created by normal IVF. So we're taking single cells from the inner cell mass to look at the gene expression in them and to compare them with embryos created by normal IVF. We're also testing for chromosomal abnormalities, and our tests so far indicate that there's no increase in the incidence of chromosomal abnormalities. We're modifying the procedure to minimize the mitochondrial DNA carryover, and we're deriving embryonic stem cells to investigate the fate of any mitochondrial DNA that's carried over with the pronuclei. So all of this work is ongoing. And now, so that's for pronuclear transfer. So what about the other options? We spoke about doing this procedure before the egg is fertilized by moving this meiotic spindle. This is called Meiosis 2 spindle transfer. This is the second meiotic division that it's waiting to do, and you're transferring the spindle, not the nucleus. So, and as I explained this earlier, so I'm just going to tell you where we're up to with that, and this comes mainly from a group in Oregon. In 2009, there were healthy monkeys born following transfer of the MET2 spindle, and here you can see there's a monkey oocyte seen through this polarized light. You can see the little spindle here, and then that's removed, and what they got in the monkey was promising. 51% of manipulated oocytes formed blastocysts, and 3 out of 9 females became pregnant, and no mitochondrial DNA carryover was detectable in the monkeys. And these are 2 of the monkeys called Mito and Tracker, after Mitotracker dive, which is used to see mitochondria under the microscope. So in 2012, the same group published proof of concept work in the human oocyte. Here you see the spindle again by this polarized light. And they removed that spindle here and then fuse it back in. So what they found in the human, and they didn't find this in the monkey, was the high incidence of abnormal fertilization. Only 48% of them fertilized normally. And it was mainly, they had these 3 pronuclei. So instead of having 2 pronuclei, as we saw earlier, they now have 3. And the sort of indication and one conclusion is that this happens as a result. So when the egg is fertilized, its chromosomes divide, and half of them are lost in this second polar body here, and leaving 1 copy of each in the oocyte. And so We think what's going wrong here is this formation of this second polar body is not happening, and an extra copy of maternal chromosomes is being retained in the oocyte in a significant proportion of these manipulated eggs. Nonetheless, when they looked at blastocyst formation of those that fertilize normally, it compared favorably with controls. So once this problem of abnormal fertilization is tackled, and I believe it can be tackled, this spindle transfer may also be an option. So just to summarize on the new techniques, pronuclear transfer and metaphase 2 spindle transfer provide promising options to reduce the risk of mitochondrial disease, and further research to optimize the efficiency and to test for safety and efficacy is ongoing. So looking at just in these final few slides, how we might translate these these techniques into effective clinical treatments. An important point is that treatment is likely to work best for younger women. So, and just to explain that, this is data from our IVF clinic, and what you see here in the purplish color is the number of oocytes, and this is the implantation. The implantation is when we replace an embryo, does it implant to give us a heartbeat at 6 weeks. So this is at 100% here at the age of 30. It's normalized to the number of oocytes and implantation rate at the age of 30. And you can see here there's a steady decline in the number of oocytes as we go in that last decade of life, from 30 to 40. And there's an even faster decline in the ability of those oocytes to give rise to a pregnancy. So I think it will be really important for these treatments to actually, for clinicians who are looking after these families, to counsel the women about having eggs stored while they're still young. And then another point will be, I don't know what it's like in Australia, but in most of Europe it's very, very difficult to recruit egg donors for treatment. And I think it would be really important to develop effective ways of communicating the difference between whole egg donation, that is donation of nuclear DNA, and mitochondrial DNA. And I think that these 2 points will really be key to turning these into successful treatments. And I'd like to finish there and acknowledge the people who've done the work. So in my lab, they're doing the reproductive biology. The pronuclear transfer has really been taken forward by Louise Hislop, who's done a great job in optimizing the procedures for normally fertilized eggs. Then in Doug Turnbull's lab, he and Lindsay Craven are doing the mitochondrial genetics, measuring mitochondrial DNA carryover. The egg donation program in the IVF clinic is coordinated by my clinical colleagues, Alison Murdoch and Meena Chaudhry, with the help of 2 research nurses. We're collaborating with Dagan Wells in Oxford to look at aneuploidy in the embryos, Kathy Neakin and Francis Crick to do the gene expression. And I also have to thank James Lawford Davies, who's a legal person without whose help I believe we could not have gotten the research license to do this research in the first place. And of course the Wellcome Trust, who funds the work. Thanks for your attention.